Hi, everyone. Welcome back to the STEM Career Talk series at Berkeley Lab. Berkeley Lab is a Department of Energy funded multidisciplinary research facility where innovations in advanced materials, life sciences, energy efficiency, detectors, and accelerators serve the country's needs to technology and the environment. My name is Sophia, and I'm an intern here at the lab for the summer with Experiences in Research program. Today, we are joined by some amazing panelists to talk about the Department of Energy's BRAVE-funded Phage Foundry project.
Hi, I'm Angelina, and I'm also an Experiences in Research intern at the lab. Sophia and I will be co-hosting along with Faith today, and we are very excited about today's panel.
I'd like to start introducing our panelists for today. First, we have Dr. Vivek Mutalik, who is a staff scientist and principal investigator at the DOE BRAVE Phage Foundry project. Now we'll be turning it to Vivek to give a short introduction of himself and a brief explanation of what the Phage Foundry project does.
Thank you both, and thank you everyone for giving us an opportunity to present what we're doing at the Berkeley Lab. I'm a staff scientist at the Berkeley Lab. My name is Vivek Mutalik, and my background is most of my education has been in India, and then I joined UC San Francisco as a postdoc, essentially to continue my research. Then I joined UC Berkeley and Berkeley National Lab around almost 18 years ago now. It's been a while I'm here. So today I'll be presenting some of the stuff that we are doing. I've made it very simple so that everybody who's not biologist or life science person can understand what we're doing at the Berkeley Lab. Let me share the slides. Yeah. So today I'm going to talk about use of viruses to fight some of the superbugs, and that's what Phage Foundry is doing. This is the scene from the hill. Obviously, you must have seen it looking at the bay. So I'm really proud of being at Berkeley Lab, and I enjoy my work every day. My lab, some of you will meet some of the members from the lab later today, is mainly focused on a couple of things. One is how to engineer a bacteria and what happens to the engineered bacteria in the environment if I release that engineered bacteria in the soil or a human gut or on plants. What are different factors influence the engineered function? So let's imagine it does some function, and how does these other competitors, some other microeukaryotes, some other conditions like pH and so on, and some of the viruses, how they impact the function of the engineered microbe. And today I'll be mainly focusing on phages, which are viruses that the microbes get infected with. So as you can see, the COVID-19 obviously changed. It has impacted every one of us, and it has taught us that we share so much together. Other than just air and water, we also share infectious diseases. This is the whole world. And these are the top infectious diseases which are emerging, and some of them are re-emerging. And some of them are, not all of them are bacterial, most of them are virus, essentially like what SARS-CoV-2 was for COVID-19. And some of them are bacterial infections, and which are really threatening the normal world order. You must have seen in the popular press and general news articles where there are infections which is spreading across the world, which is for multi-drug resistance. What does that mean? That means these infections, when happen to humans, they cannot be treated with any kind of drug. That becomes very dangerous because then person, like a simple cut, well, the person is either cutting a vegetable or something, that cut cannot be cured or treated with antibiotics. So one of the recent example is this one where artificial tears, many people use it in their tears, was infected with one of the drug-resistant infectious disease organisms called aeruginosa. And now this person either loses the eye or sometimes in cases, severe cases, might lose life. And so this is spreading across the world. It is not only limited to some of the countries, but this has been going on for a long time, actually. How does this resistance spread? It has been spreading because we've been using some of the antibiotics. We feed antibiotics, small amount of it to animals because they can put on weight after eating antibiotics. And these taking antibiotics will kill the sensitive bacteria, obviously, like any infection. But there are some bacteria will survive and they will multiply. And when you use this bacteria, which is resistant to this antibiotics, get enriched when you put it on, animal products, so like a meat manufacturing or, produce manufacturing or preparation of food. And these animals also, release these microbes into the environment, and that can also run off from the river or a lake. So the spread has been going on for a long time, and we know about it, and that is causing this rise of superbugs. So World Health Organization and CDC has put together what are the top urgent and emerging threats. These are really scary ones. When the infection happens, whether it is in meat industry or agriculture or a poultry or aquaculture or in hospitals, you go to the hospital for general surgery or something, you get infected with one of the superbugs, and that is the reality of it, and we don't know how to treat it. And so the hospitals are connected, we are all connected, the community is connected to all of these things, right? And then the spread becomes very rapid and across the world. It's not insulated to one country. It can happen everywhere. So some estimations, like by 2050, it is possible if we don't do really take steps to stop it, it can really, the multi-drug resistance can really surpass cancer in some of the world parts. It obviously impacts our industry, impacts our healthcare systems. It can impact economy and so on and so forth. It is not only all of these things, but many of folks will lose life on a simple thing like an infection. What are the solutions, right? Solutions are, that we can take drugs, we can take vaccines, these like medicines and vaccines. There are some other possibilities, there are solutions happening. We will be talking about these viruses, which can be used to treat these kind of infections, okay? And all of these are all in the pipeline for development. So what are the viruses, right? Phages. These are phages or viruses. These infect only bacteria, so they don't infect humans. It looks like a lunar lander, essentially, and I'll cover I'll give you an introduction of what is this particle looks like. It's non-living particle, but becomes functional when it goes inside the bacteria on its host. The studying this can open up so many avenues, for that is what is exciting to me and my group. By studying these phages, we not only understand who's there in the environment and what are they doing, but we also we can use them for a variety of biomedical applications or environmental applications, whether developing vaccines, developing diagnosis. We can do a lot of applications like imaging and so on and so forth. So this particular phage virus can become a platform for a variety of biotechnology solutions. Where do we find them? We find viruses wherever we find microbes. So you take a small amount of soil or a small amount of water, it's filled with millions of viruses. On animals, on humans, any part of anywhere you find microbes, which is essentially everywhere on Earth, you will find these viruses. And they come under, like us, they come as a totally different structure. Some of them have a nice, fancy, star-like structures on their tail. Some of them are fancy, covered with some other structures. These are all interesting viruses. They are some of the images. These are microscopy images that one can take when you culture them in the lab. And some of you have been taking this course, which we are taking here for phage isolation and studying them. And this is what they will be studying in some of the modules. So how does this infect? So imagine this lunar lander kind of a thing, which will sit on a bacteria. This is my bacteria with its own genome, and it sits there, and like a syringe, it injects its DNA or genetic material into the bacterial cell, and then it makes copies of itself, and it hijacks the whole machinery of the cell. It makes more copies, and essentially, end of the day, end of the cycle, it kills the bacteria and releases hundreds and thousands of virus particles, which will release into the environment, and they go and kill other bacterial cells. So this interaction is very specific. That means there is exactly one phage for that particular bacteria. There is a recognition. It's like a lock and key. And so the bacteria is also not naive. It also fights back. It also has this different roadblocks for this infection to not to process, So there is a whole, it's called phage cycle. That's what it is. And essentially, and phages have been discovered a while back, almost 100 years back, they have been discovered. And this is some timeline of different type of phages and different kinds of phages. Phages have been thought of using as a therapy for a long time. And here is some of the examples. I'm going to touch upon some of the instances where phage has been used. Here is an example where this is Stephanie and her husband Tom, and this is a kid from UK who got infected with one of the really drug-resistant Mycobacterium abscesses. Tom got infected with one of the nastiest pathogen called Acinetobacter baumannii in Egypt, one of the trips they went. And there is a TED talk, there's a YouTube by Stephanie about the story of this person, how she mobilized hundreds of thousands of people and saved his life because he was in coma because of this infection, because he was not able to get treated with anything. Same thing with this to a kid who got infected with this bug and cannot be treated. But the phages have been used in these cases and they have been super successful. And this has been a role model. Tom has been role model for understanding that phages can work. So, but we do not understand why some phages work in some cases, but do not work in some other cases. So there is a challenge here. So the way the phages have been used is you take an environmental sample and you isolate phages. They will look like this. Essentially, this is a lawn of bacteria. On a lawn, you see the clearance. That means this virus is killing that particular region of a bacteria. So the phage isolation is done, and some of you have done in the lab, in the Berkeley lab course. Then we purify them. We do the genome sequence, so we understand what is it made up of. Then we test them across different bacterial strains. Then we make a cocktail, essentially mix a bunch of different phages so that the bacteria doesn't have chance to fight back. So imagine having 3 different phages to kill one bacteria type, right? But there are a lot of challenges to this cycle. You spend millions of dollars doing this work. It's not a scalable object. And why it is not scalable is the global challenge of this antibiotic resistance is non-trivial. These are all top biothreat or multi-drug resistant pathogens. Each of them are like a multi-million-dollar expense on our healthcare and human health and so on and so forth, as I mentioned before. So if we want to really scale our approaches to treat all of these infections, then we need a systematic approach. And that is what my team is doing here. The Phage Foundry is an idea and the platform. It's a national capability to bring together a lot of diverse expertise and technologies to build largest collection of phages on a top pathogens so that we can perform and study them. We can make formulations and use them in applications. And the interesting thing for us also is can we really predict which phages or which viruses to use when there is an infection that comes up. So imagine there is an outbreak happen somewhere in the world, and we will, because of the genomic sequence, you can sequence the DNA or the information of the genetic material. And then the question becomes, can I really formulate the treatment just based on the genomic sequence, not even testing it? And this is what happened in SARS-CoV-2, right? The infections they rose in China, but then US already had a vaccine ready based on the genomic material of the virus. So can we do that, something similar for bacterial infections? So for doing that, we need data, right? Machine learning, ChatGPT is not going to give me a solution because ChatGPT doesn't have this kind of a data. So one of the key goals of Phage Foundry is to establish a platform where we will generate this kind of data. Now the question becomes, how much data is enough for making this prediction, right? We don't know how much data. We don't know what type of data. We don't even know how to really start building this kind of a data sets where genome sequence of a bacterial strain can connect to its countermeasure, whether it is using phages alone on their own or phage and antibiotic combinations, you know. So one of the simplest thing in our lab we do is we try to rapidly evolve. That means we isolate a lot of phages. Then we try to evolve them. That means we try to create mutations or changes in the viruses. Or we also use engineering principles to engineer these viruses or phages so that we can create variations and then we study them. And then so that we can have these kind of countermeasures for the antibiotic resistance. So this involves a multi-step process. I'm not going to go through it, but it involves like a lot of team. We have about 15 different labs working on this along with us, around 25, 20, 30 researchers working on different aspects of it, like isolation, characterization, understanding their evolution, understanding the machine learning algorithms. Obviously, it is not one team effort. It is like a multiple teams, and that's what national labs do. You know, it's always a team science. So a lot of teams come together to try to solve a problem which is of a global nature. Not single PI or a principal investigator or supervisor can do this kind of research. So you will hear today, this is a research team which is in the Phage Foundry, which has been part of the workforce development program that Faith and other team has been running on. So this is you will hear some of the researchers and where they're coming from and what are their interests. This is the entire team that is being part of the BLDAP workforce. So maybe I'll stop there and then happy to take questions if you have, or we can wait till at the end.
Thank you, Vivek, for giving us more context about what phages are. I'd like to continue introducing the rest of our panelists for today. This morning, we are joined by project scientist Dr. Dennis Pia, research associate Mohammed Alayuni, and a student researcher Madeline Svab. We'll have our panelists tell us a little more about what they do at the Phage Foundry, starting with Dennis.
Hello everyone, I'm Dennis. I'm very glad that I got to be a part of these STEM series, and I'm very excited to share my experience with all of you. If you have any questions, feel free to put them in the chat and we can talk about that later. I'm originally from Nepal. It's a small country which is bordered by China on the north and India on the east, west, and south. And one of the things Nepal is very famous for is Mount Everest. It's the tallest peak in the world. And I grew up in a small town which was located on the top of the hill. And from the hill, we could see this beautiful mountain ranges every morning. And on the right, I have the picture of the town as well. It's a very quiet and very peaceful town. Next slide, please. So I did my PhD in biochemistry from Texas A&M University. Within the biochemistry department, there is a Center for Phage Technology, and it's where I got introduced to bacteriophages, and I have been working with bacteriophages for most of my scientific career. After my PhD, I joined UC Berkeley as a postdoc. I was working with Adam Arkin and Vivek with phages again. After my postdoctoral training, I ventured in the biotech startup world briefly, where I was working with RNA therapeutics. And after my time in this startup, I came back to Berkeley Lab as a project scientist, and I'm working with Vivek in the Phage Foundry project. Next slide, please. Today, I will briefly talk about one of the projects that I am leading in the lab. In this project, we are trying to identify if any phage sequences are inhibiting other infecting phages. As Vivek talked in his intro slide, these phages can be used as a therapeutics, but to be able to use them as a therapeutics, we have to understand what other inhibition mechanism can this bacteria have. And as Vivek mentioned, wherever we find bacteria, there are phages. And in some cases, these phages are present as in a temperate life cycle where they are not producing active virions, but they are integrated into the genome of the bacteria, and they are just replicating in the DNA form. So in the schematics in the left, it shows that whenever there is a P1 lysozyme, this uh, classical E. coli phases like T3, T7, lambda, and P1 will become inhibited by that P1 lysozyme. So in this project, we are trying to identify what gene elements are present in these P1 that's blocking the infection of these phases. So for these, I am applying CRISPR-Cas system, where I'm inhibiting a particular gene at a time. And if a particular gene is playing a role in preventing the infection, then when we repress that particular gene, then these phages are able to infect that particular host. And this kind of information is very crucial because like this is like an unknown world, and having all of this information will help to develop the machine learning model, which will eventually can be used to predict the phase host interaction. Thank you.
Mohammed, you're mute.
Yep. Hello, everybody. Thank you for your opportunity, for this opportunity and the time for me to talk to you. And I'm happy to share my experience of what I'm doing here at the Phage Foundry. So just a background, I've been living in Virginia, and I moved here to the Bay Area in January of this year. And when I was in Virginia, I went to Harrisonburg High School, which is actually located in a very small city called Harrisonburg, which has a very unique university called James Madison University. After I graduated, I moved more north in Virginia, which is where George Mason University is, which you can see at the top right. And that's where I got my bachelor's degree, and I got a bachelor's in science, and specifically in biotechnology. I was also working in this about a semester long class called C phages, where basically we were collecting phages from the environment and trying to isolate phages that are specifically able to infect a non-pathogenic version of tuberculosis called Microbacterium smegmatis. And then after that, when I was about to graduate, I actually started to work part-time at a cancer therapeutic startup, which is located in Maryland, called Remedy Plan Therapeutics. And over there, I began as a research assistant and eventually made my way to a research associate. During my time at Remedy Plan, I was also studying part-time as a master's student at Johns Hopkins University, which you can see at the bottom right. That's like a picture of the campus. And while I was also studying over there, I did do some, I actually developed a nonprofit called Nova Sci-Fi Labs. And Nova is basically the area, Northern Virginia. And basically, this is a nonprofit that's focused on teaching STEM to anybody who's interested. So after I was done with my master's at Johns Hopkins, I decided to move to the Bay Area, and that's where I started to work at Lawrence Berkeley National Laboratory. Next slide, please. So here at the Lawrence Berkeley National Laboratory, I'm glad to be working at the Phage Foundry. We have an amazing team, and my focus is really on Pseudomonas aeruginosa at this point. As Vivek mentioned, that we do have a lot of different goals, but at this point, I'm focusing on Pseudomonas aeruginosa, and this is a multi-drug resistant pathogen, and I am focusing on isolating phages that specifically are able to infect it and kill it. So that entails collecting environmental samples, isolating the phages, and then also preparing the phages and then sending them for sequencing. And then once we get their genomic sequences, we would send that to a bioinformatics team, and they would annotate the sequence and basically send it to the database where it would help us generate this machine learning algorithm. Another thing I support with are our mouse studies. So I do prepare Pseudomonas aeruginosa and a certain phage that is able to infect it, called LIT1. And when we're doing the mouse studies, I do prepare those materials and provide it to the research team that's doing those studies. And then on the side, I'm also looking at other research projects to work on. And that includes this interesting project, which has to do with using C. elegans as as a model organism to test the efficacy of different phages on basically treating C. elegans that is infected with Pseudomonas aeruginosa. So that's it for my end.
All right. Hello, everyone. Thank you so much for having me today. It's a pleasure to be here to talk to you all. My name is Madeline Svab. I'm an undergraduate researcher who works with the team here to study phages at Berkeley Lab. I attend UC Berkeley, where I'll be going into my third year of studies coming this fall. I'm a simultaneous degree student, so I study not just microbiology, but also biochemistry at the university. Next slide, please. So I did not grow up in the Bay Area, but I did grow up in California. I was born in Los Angeles, where I attended John Burroughs High School. When I was in high school, I really had no idea what I wanted to do with my career. I just knew that I really liked chemistry and that I really, really liked microorganisms. So I learned what a phage was for the first time actually when I was in high school, and I was just immediately so interested in them. I wasn't planning on trying to become a scientist, but I love science so much that with some encouragement from a really excellent teacher that I had in high school, I decided to go to school to study biology. And then I began attending UC Berkeley, where I eventually became an undergraduate researcher in the lab of Richard Harland, where I worked to study xenopus embryos, which are frog embryos, if you don't know. And I did embryonic microsurgery and some other work at that lab. But I knew I still had a really, really deep interest in microorganisms. And that's when I found out about Vivek's lab here at Berkeley Lab. And I was just so interested in the research being done here, especially since I had been interested in phages since I was in high school. So I applied to work at the lab, and shortly after I joined the team in May of last year. Next slide, please. So I have a lot of different projects that I'm working on here at the lab. So I'm going to just really quickly touch on a little bit of what I'm doing here. So when I first started working at the lab all the way last year, I first started working isolating single-stranded RNA phages, which are a type of phage that are relatively poorly understood and also play key roles in environmental systems. So I did some work isolating and studying those kinds of phages. But I pretty quickly moved into starting to work with phages that infected specifically strains of E. coli. So a lot of my work revolved around doing large experiments that produced tons of data about E. coli phages in order to help predict how phages were infecting E. coli. So a lot of the work that I do here right now at the lab is helping to maintain a library of almost 200 phages that infect E. coli that we use here to study that infection at the lab. Some of the other work that I'm doing includes helping to build new genetic libraries called DubSeq libraries to study phages. And more recently, I've also begun to study something called bacterial talosins, which are like a toxin that bacteria produce that kind of look like bacteriophages, which is a little bit confusing. But they help us to understand bacteria better and how bacteria live in communities together better. So that's work that I have been interested in doing recently. So I have really enjoyed getting to work here at the lab. I find phages just so interesting and worthwhile to study. But additionally, I feel really lucky to be able to work alongside people like Vivek, Dinesh, and Mohammed, who are so willing to help me learn. So it's great to be here.
Thank you for sharing. We'll now be moving to our Q&A section of the program. I'd like to encourage our audience to ask some questions in the chat box while we start off with some questions you've created. So the first question is specifically for Vivek. What is it like juggling doing research and leading a team of researchers?
That's a great question. Berkeley Lab, as we mentioned, it works on mainly on the team science. Essentially, we handle or try to solve problems which are so global that one single PI cannot do or single research team cannot solve it. So one of the first things that starts with is finding a global problem, which obviously there are so many of them. Obviously, and that most important is to identify which problem resonates with you, what kind of skill sets will help in solving that. So because I'm a scientist, I'm passionate about science and work on the bench. I really do experiments with my own hands. So important is to have a team of scientists who are also passionate like me, and that you heard some of the members of the lab presenting their stuff today. So it is like a small business. Running a lab is like a small business where everybody has their own defined, their own goals are defined, their projects are defined. And there is a fun element to it, right? You have a problem to solve and you know how to solve it. There are steps to take. Some innovation need to happen. So my goal and my role in the team is to provide funding for when you need funding for science. Obviously, we want, one of the other things that I do is to write grants, essentially to raise money. And then second one is the motivation to keep the team motivated and keep it, inspired, because innovation happens only when everybody is comfortable in their own self. So to provide that peaceful environment, and also my other role is to insulate the team from other pressures or managing other things on the science aspect. So one of the challenge in handling a team and then also the scientific goals, everybody has their own interest. But at the same time, we want to do innovation in science. So excelling, asking everybody to do better than what they are doing right now. So creating that environment is what, is fun part. And then finally, doing science is not just doing experiments and getting results and publishing those results, but also, keeping humility, being sensitive, being empathetic to the environment and surrounding colleagues. Some of the things that have really helped me in doing all of this work, obviously having a fantastic team, that's the most important part, because ideas can come and go, but the team is the core importance. And then keeping myself, how do I keep my motivation and inspiration alive is I spend time with myself more, do some meditation. I spend a lot of time in spirituality, and it helps me to center myself so that what is most important in life and how do I help with my skills to the world. So that's how the whole science that I do in the lab works.
That sounds like a lot to handle, but your dedication to your team is very commendable, so thank you.
Thank you.
For our next question, I'd like to ask Dennis and Mohammed, what is the difference between conducting research at a national lab versus industry or a college and university? Also, at this time, I'd like to invite the full panel to turn on their cameras for the rest of our Q&A session.
That's a very interesting question. So far with my experience, I haven't found much difference in the research conducted or like the work in the university versus the national lab. But when we compare the research being done in either like the um, industry side or the academic side, there are some differences. For example, in the industry side, it's mostly focused on like product development. Eventually, people do some science, but eventually it's geared towards making a product and it has to generate some profit at the end. Whereas in academia, it's not always the case. Like we do science, but we are trying to like understand the basic biology, or in other field, like trying to understand the basic things like which are driving certain phenotypic mechanisms. And when we compare that to like phage research, for example, in academia, like we are trying to understand how like how like different genes present in the bacteria make that particular host resistant or sensitive to a particular phase, right? Whereas in case of industry, the research will be mostly focused on what phases can kill a particular bacterial host. And if a phase can't kill a bacterial host, they'll just like move and work on different phases.
Yeah. For me to add to that too, from my experience, I would say from working in industry in a startup and then working at a national lab, I would say one thing I realized is that a lot of the focus is on the people themselves. So the people who are conducting the research in the national lab, it seems like a lot of the focus is to promote them, to promote how the team and promote the people who are working here, rather than in industry where a lot of the focus is on the product itself and how like this company is making the best product or this company is making the best services. While in a national lab, it feels more like we have the best people and we're promoting the best people. Another thing is that in industry, I felt like it was much more focused and fast paced. So everybody's like focused on a small thing and it's that's all they do. And it's really fast paced so that they can execute and get the product developed or get the service going. While in a national lab, I've from my experience been like it's focused as well, but also there's free time to kind of investigate things that you're interested in. And then another thing, since I worked at a startup, I would say, and it was a relatively smaller one, a lot of the people who I was surrounded by have been mostly people who are like relatively like they have a PhD and they're kind of like mostly done with their studies. But it seems like in a national lab, there's a variety of different backgrounds. There are some undergrads, there are some grads who are just like all they have is a master's, and then there are some postdocs, and then also people who have PhDs. So it's like a variety of different people with different educational backgrounds. So that's something I wanted to add.
Yeah, that's really cool. Thank you for sharing.
Sure.
Now, this next question is for Madeline and Vivek. So how do you both deal with setbacks, failures, or having to readjust your goals for the project? How do you keep motivated?
I think that one of the biggest things for me when I was starting to do science was kind of adjusting my expectations. I don't expect a project to work the first time, basically ever when I'm doing it. I don't expect things to be easy. And because I'm always expecting those setbacks when things do go easily or work the first time, it's always kind of a pleasant surprise versus, what I was expecting in the first place. But generally, yeah, I expect things to take twice as long and be twice as difficult as I initially kind of always anticipated a project to be. And that definitely helps to some extent. That's something that I think I always try to keep in mind when I'm working on projects. But in a much more real way, I think that I choose to take failure and use that as a motivation to continue working on projects because I'm a very problem-driven person. I like to continue to investigate problems. That's why I do science. So for me, whenever failures or setbacks come up, that's just another reason to continue looking into a problem. And I found a lot of things that I would have never learned or known previously from failures and setbacks. So I know it's kind of cliche, but I think that it is true that you learn the most from failing. And also just kind of like a last thing is just, like consulting with other people and using the people around you as a resource and using them to troubleshoot and also just to get new perspectives on the work that you're doing and how you can improve it. And just, yeah, like just learning from the people around you.
Yeah, just wanted to add that, I like what exactly Madeline was saying. Most of the experiments that we do usually fail because there are a lot of reasons, technical challenges. And it's very difficult sometimes, just to be clear. Sometimes when you spend a whole year on particular project, you're thinking about it, you're reading, you spend a lot of time, dedication of the time and effort and funding towards one particular problem. And then at the end of those experiments or end of that project, you realize that this is not going to be solved or it is difficult to solve because of some challenges, technical challenges, or you can't really measure that particular thing. So it is disheartening sometimes, you obviously you lose hope. You said, oh my God, I spent so much time on this and then it did not work. It's easy to get frustrated. So sometimes you just take a break, you go home, go into the nature or spend time with your family, you do whatever you like to do, and then come back and revisit and think whether it is going to be pursued or you want to drop it and then go to the next challenge, because there are so many things to do anyway, right? There are so many interesting ideas that you want to follow. So one way is to get frustrated and lose hope. The other one option you have is, frustrated, take time, and come back with force, or come back with new energy where you can go and do better than what you did before. So it's a skill. Sometimes it takes time to get back, but you will eventually realize that is the only best path for you to keep yourself excited about life and then motivated about solving a problem.
Well, it's great to hear that you both use failure not to discourage you, but to motivate you. I think a lot of our interns will definitely relate to that as they experience challenges on their own projects.
Our next question is for Vivek again. So, phage therapy is currently not a licensed treatment in the United States, but the therapy is used to treat bacterial infections in other countries. Since it has recently experienced a resurgence, do you think this treatment could be part of the future of modern American medicine? And why do you think this therapy has made such a comeback in the West recently?
Wow, that's a great question. A couple of points. The phage is discovered 100 years ago, as I mentioned. Some of the countries like Soviet Union that time, and then some countries around that, they have been using phages, even though antibiotics was discovered. They have been continue working on them and archiving them, storing them, studying them, using them in a clinical practice. In the Western countries, when the antibiotics was discovered, everybody went to antibiotics during the World War because the manufacturing of these antibiotics was easy, simple. Also, the application was very straightforward. You can make a pill, you can send it, so it's much more easier. Whereas now recently, suddenly we realized that the antibiotic resistance has been really ramping up, and it is really coming up into any of the hospitals that you might have visited. For example, in the hospitals in the Bay Area, often find infections cannot be treated with any antibiotics there in the hospital. So in those situations, suddenly Western countries realize that, okay, now this is a real problem. We need to solve it if we want to really not let this get out of hand. So of course, it's not licensed, it is not in a clinical practice right now in a mainstream, but when there are no options, there is something called compassionate usage. So let's imagine a patient in the hospital not getting treated with any of the antibiotics. Then the patient can, or doctor can request, or the person's family can request FDA to allow a compassionate usage of phages to use for patients. And that has been going on for some time now. So there are some success stories, some of them failures, and there are a lot of researchers working on understanding the challenges. Now, whether it is going to be a mainstream or not, that's an interesting question. So in my personal opinion, phage by themselves may not replace antibiotics because antibiotics are so powerful and so invaluable for human life that replacing that with something phage may not happen. But what we are realizing, and researchers in this field realizing, that it would be nice and smart to use phage along with antibiotics as a combination. So let's imagine a person is insensitive to antibiotics or resistant to antibiotics, but then we can give phage, and then combination of that can be used much smartly. And so in my personal opinion, it may not become a mainstream drug, but it can be a supplementary or maybe additional adjuvant, which we can use it with antibiotics very rationally. That means thinking about it, how to use it, so that more research is needed, and that's what the Phage Foundry is focused on.
Yeah, that sounds like some very exciting new areas of medicine to study in the future.
Yeah,
Thank you.
great.
Okay, so our next question is for Dennis. So how do you know which phages are the most suitable for treating certain types of bacteria? After the phages destroy the bacteria, where do they go?
Very interesting question. As we learned earlier, these phages are like everywhere, and it has been estimated that like for every cell of bacteria, there are 10 viruses around. But it's not necessarily true that all of those viruses are infecting the same bacteria. So the first thing that we have to make sure is the phages that we are selecting can infect a particular bacterial strain, and that can be done in a lab setting by plating a bacteria on a surface of a LB plate, and we can spot like phages on those. And if the phages are able to infect that bacteria, then it will create a zone of inhibition, which basically means that every time a particular phage finds a bacterial cell, it will just infect the cell and kill the bacterial cell. That way, the bacteria cannot grow wherever the phages are present. So that's like the first metric. And second metric is we will sequence these phages, and we have to test these phases like if they are encoding for certain genes. there is a specific kind of life cycle for some of these phages where once they inject their DNA, it doesn't replicate, but it will integrate its DNA into the host bacterium. So we have to rule out that phages are able to do that. And for that, like we can look for like certain signature genes in the phage genome. Because if the phages are able to inject their DNA into the cells or like integrate their DNA into the bacterial cell, then it could impart like certain advantages to the bacterial genome because the phage genomes could carry like antibiotic resistant marker, or it could carry like certain like toxins, which could even make those bacteria like even more harmful than it was previously. So that's like the second metric that we have to be very careful about. And furtherwise, there are certain like stability factors that are important as well because these phages should be stored at like optimum temperature, but and then only it can be used. Otherwise, they would like degrade over time, and the active particles in the formulation can drop down to like zero if those are unstable. But again, like that part of experiment takes like a very long time. And as we mentioned, like phages are considered as like the last resort treatment, and in some cases of like compassionate use, there won't be any time to rule out all of these metrics. So people just use whatever active phages that they have at that case. And for the second question about where the phages go, so these like viruses are considered as like the borderline between the living and non-living things. So as long as they are inside their host, which is the bacteria in the case of phages, they can like actively replicate. But at the end of the life cycle, like they will kill the bacterial host and they are released in the surrounding. So in the lab setting, that would be like the growth media. Whereas in the nature, like it could be like water where this bacteria are found, or like the soil environment where this bacteria are found. So like once it's released into the surrounding, then at that point, like it acts as like a non-living being, so it cannot do anything. And all of these, since these phase particles are composed of proteins and DNA, they will just stay inactive at that time. And as soon as it find its new host, it will start the new infection cycle again.
Okay, thank you so much for sharing. That was interesting.
Our next question goes to Madeline and Mohammed. So what drew you to working at the Phage Foundry?
I can go ahead. And so for me, when I was actually at George Mason, I was involved in this course, which was part of the C phages. And in that course, as I mentioned, I was isolating phages from environmental samples and trying to find which phages are able to infect Mycobacterium smegmatis. So I just realized during that time, it was a very fun experience for me because it was, I had the opportunity to go out in nature, collect samples based on what I found interesting, and also then bring them back and start working with these samples, preparing the samples, isolating potential phages that are found in them. So all of that, I think, is really something that I found interesting because I really like going out in nature. I like going on hikes. I like going to just going on right by rivers or on boats. And so I do have that like naturalist perspective that I do appreciate, and I really try to take advantage of that whenever I have the opportunity when I'm working in biotech. So, working at the Phage Foundry and specifically working with phages really gave me that opportunity to make use of my naturalist perspective, where because I do like going outside and collecting environmental samples. So that's one thing that drew me. And then also the potential of phages, I think is phages do have a lot of potential in biotechnology. There are various different applications that I do think have not been tapped into yet. And that's something I would love to learn more about, and also potentially use these applications and develop new ones as well. So that's what drew me into the Phage Foundry.
Yeah, I resonate with a lot of a lot of what Mohammed said. Like I mentioned, I was really interested in phages and viruses in general when I was younger, and just like how they exploit biological life for their own gains. But I was also just very interested in how viruses could be applied to help people and how studying viruses could do good things for people around the world. So that's why I decided to just take the chance and apply to work at the lab because I was just so interested in the work that was being done here. But the reason why I've stayed here and why I've liked my role so much is just because of the environment of collaboration, getting to work on a topic that's just so interesting. So for me, it's a combination of factors. It's the subject matter, it's the actual ability to study viruses, but it's also just the people and the types of collaboration and the work that's being done here and the way that it's being done.
Yeah, that sounds really cool. Thank you so much.
Okay, now this next question is for Vivek from the audience. You mentioned that one of the better options is to use antibiotics and phages together. Is there any fear of virus outgoing both viruses and phages? Is it even possible for a virus or bacteria to evolve to such a level?
Yeah, that's a super interesting question. Yeah, no one has seen such super virus coming out of, virus and bacteria fighting together. They have been fighting this war for a long, long time, thousands to millions of years. They have been in the environment. Bacteria, microbial communities are always there, and virus communities are always there. They're fighting with each other. So no, we haven't seen any of that. For more than 100 years of research has been done in this area, and there is no such thing as a super virus that will overcome bacteria or virus evolution. So that they are evolving to survive and infect, and bacteria is evolving to fight the virus infection, like what we are fighting with virus infection. Like imagine we all are registered to some of the early versions of SARS-CoV-2, similar. The bacteria also are evolving. So the simple answer is no. There is no such threat, and no, these are all safe viruses. These are all, and it has been used on humans before, and we haven't seen any such instances.
Well, thank you. That's very encouraging to hear that.
And our last question for today's session is, this goes out to all panelists. What do you wish you had known in high school or early college that would have made your career path a bit easier?
Maybe I'll go first. This is something really, really I think a lot when we go and teach this module with all of the students, some of the students that are on the call today. I really get motivated. I see the spark in their eyes when they can see this phage on a plate, and then, wish I had done this in high school. I did my high school in India, which has totally different environment and funding and all of those questions. But just exposure of this kind of a science that Faith and all of the team that is being done here at the Berkeley Lab is just so inspiring. So I hope I had this kind of an environment. I'm so glad that Berkeley Lab is enabling this kind of experience to all of the audience today. These students, it is such a privilege to getting training, and finding what you like is such an important aspect. Anyway, I'll stop there. I can talk about it all day.
Maybe I can go next. Same as Vivek, when I learned about biology and chemistry in my high school, it was mostly based on book knowledge. So I had learned about this bacteria and viruses, but it was only based on pictures on a book. I have been working on phages, but the first time I saw a plaque on a bacteria alone was in my first year in grad school. And the first time I was able to see the image of actual phase in under a microscope was maybe in the second year of my grad school. So it had been so fascinating to be able to learn and do all of those things in the beginning year of my high school. So if I was able to do all of those hands-on experiment at that time, I think I would be even more compassionate and more passionate about science.
I can go next. So one thing I would recommend is really thinking more and spending more time thinking about the thing that you want to do long term as a career. because I felt like, the more you think about anything, of course, the more you're going to find answers to. And so, especially when it comes to your career, it's something you're going to do your whole life. And you do spend most of your time in your life working at your job. So usually it's like a 9 to 5. So that's Monday through Friday. You get the weekends where you could do other things, but otherwise, most of your time in the week is spent on your job. So it's spending that time is really going to pay off when you start figuring out that this is something that you like to do, and when you start answering these questions. Because if you do, you're really going to live a more inspiring life, in my opinion, when you're working in something that you enjoy and you go to work and you enjoy what you're doing. Another thing is that I would say connecting with your teachers, connecting with your professors if you're, in grad school or as an undergrad, because actually my first job when I was working at the startup, I got that because of the teacher who was teaching me a virology class, and she told me that she has a startup, and I kept on talking to her, and she was telling me that once you graduate, you can just reach out to me, and we can, potentially you can find a job at, at the startup that she's working at. So definitely connect with your teachers, get to know who they are, get to know like what are they doing besides teaching, and that could be an opportunity for a job later on, or just like an internship. And that's another thing too, is like if you think there's something you want to do, like a certain field, if you're interested in biotech, if you're interested in, bioinformatics, really get your hands dirty and start doing that as soon as possible, because that's going to give you that time and that experience to know if this is something you want to do long term. So find organizations that are doing things that you like, and get involved with them. So getting experience is really going to take you a long way.
Yeah, I think that one of the most important things that I would have to say is just that it's okay to not know exactly what you want to do right now. A big part of figuring out what you want to do is figuring out what you don't want to do and trying lots of things and getting to say, hey, this isn't for me, or hey, this is really for me. I really like this. So don't be afraid to just try anything that seems even a little bit interesting to you. And if you don't like it, if it doesn't work out, that's okay. That's part of it. That's part of learning what you want to be doing and getting to the place where you're supposed to be. Yeah.
Yeah, thank you for all the good advice, especially for incoming college freshmen and also high school students as they continue on. But now I'll pass it to Sophia to end the session.
Okay, thank you guys all for your kind and encouraging words. It was all very inspiring, and I'm sure a lot of our interns will take this into their future and try to adopt these same habits to their career. I would like to remind our audience that our next career talk is on quantum research on July 19th, and we would really love to have you all there.
Thank you.
Thank you. Good luck,
Thank
everyone.
you, everyone.
Thank you, everyone.